if you are uncomfortable saying anything, feel free to just put in the chat and we will read it out and answer that way. If you're comfortable talking in front of people and being recorded, then just go ahead and turn on your mic and ask us the question verbally. And anything else I need to cover, Jessica? Sounds pretty good. Awesome. So I am going to kick it off and we're going to start this. Yeah. Okay. So information section. So we're going to talk about this time is this paper that was published in Scientific America. And it's pretty much, how do your genes play into you getting the coronavirus or you getting sick? Because we're hearing a lot of people are becoming asymptomatic, nothing really is showing. You have some people who are getting very, very sick. And we're going to look at, hey, what's in your genes that's actually causing this? And this is what the article will look into. We'll do a brief talk about stuff that kind of plays into it, and then we're going to go on to something a little different. We're kind of going to branch off from the article and kind of talk about how one of these experiments might be designed. So we're going to do something a little different, but I think it should be pretty neat and informative. So let's just start off. So what we're going to talk about is the difference between the men and women with this. So as you probably have heard in the news, men are getting a lot sicker at a higher rate. I think in the article out of France, it came in that 73% of the actual severe cases are in men. So men are getting this a lot more. And this has to do with the receptor. So there's this toll-like receptors, and what they do is they are in your genes, and what they do is they are responsible for innate immunity. So if you have some of these, you are immune to certain type of things, or more likely to. Okay, so COVID-19, it activates this receptor known as TLR7. Okay, so one of these is just different. And what this is this is the one that gets activated. So if you have these, you are likely to be less affected if you actually get COVID-19. Now, the fun thing about this is it's actually on the X chromosome. And if you remember from biology, if you've ever taken it, females do have 2 X chromosomes, men have an X and Y. This means women by nature will have twice as many of these receptors, which means they are more likely to be immune or have less symptomatic from this. So this is something we found out. There's also a lot of other cool stuff. If you actually start reading into TLR7 and you start reading into it and you look at the medicines that are coming out for this, you see a lot of neat connections and why certain drugs are being tested. Sorry, that's just a side thing. But it's not ferret related this time. My sidetrack. Okay. So that's one thing that's happening and one thing we found out. And looking at the genes, we figured that out. So another thing is people seem to be getting this more often. And this is all to do with a receptor that actually needs to be binded to for COVID to actually infect you. Okay, so this is a receptor, it's the ACE2 receptor. And what happens is there is a hole that fits perfectly for it. It kind of connects and it binds to that location. When that happens, you'll get the infection and you'll start getting sick. Now, what they found is with this ACE2, there is a mutation that's known as ACE1D gene. And if you have this ACE1D gene, you're actually going to express the ACE2 less, which means you have less places for COVID to bind. So if you have less of these binding sites, you're less likely to actually get the infection because there'll be less things for the COVID to actually attach to. So with these, this is actually a neat study that's happening, and they're finding that this ACE1D gene is kind of spread out and different depending on countries. So if some countries are getting it less, it could be that this thing is in their gene pool. So that's why they seem to be getting less effective rates. Yeah. So that's the underlying kind of ideas and the genes that kind of play into it. And now I'm going to hand it off to Jessica to talk about how tests for this would actually work.
So some of the stuff that Ryan's talking about already comes from a question that a scientist has in reference to a gene that we already know about, right? So for instance, with the ACE2 receptor, it was clear from very early on that the COVID-19 virus binds to the ACE2 receptor. So it seemed really logical from the beginning to start looking at the ACE2 receptor in humans and see if there's variability, and if that variability could actually explain why some people get sick and other people don't, why some people carry the virus and viral load and other people don't. But there's a bigger question here. And that bigger question is, what makes some humans symptomatic and other humans asymptomatic? So in order to answer a big question like that, we have to do a bigger survey of the genome. We're not going to just look at one gene at a time. We're not just going to examine toll-like receptors or ACE2 inhibitors or blood groups and individually examine those genes, but rather to get a bigger picture, especially if more than one gene is involved with expressing symptoms, you want to look at the whole genome. So the question is, how do we do this using bioinformatics? Because bioinformatics is supposed to make it easier to survey the genome. And there's a certain type of study design that we would use in this certain case. And it's called a genome-wide association study, or as we short form it to, it's called GWAS. So I wanted to walk us through this because this is exactly what's happening worldwide. It's a concerted effort around the world to try and answer this question: what makes some humans symptomatic and others asymptomatic to COVID-19? So we always start with a research question, and from a research question we develop what's called a hypothesis. And the hypothesis in this case is differences in genetics underlie whether somebody is symptomatic when infected with COVID-19. So the study design looks like this. Oh, you're the one controlling the slides there, Ryan. So next slide. Thank you. So let's say, for example, you've got myself and you've got Ryan. Say hi, Ryan. Well, let's say that both of us are infected with COVID-19 and I'm showing lots of symptoms: difficulty breathing, I've got a sore throat, I've got a high fever, but Ryan's walking around and he's got no symptoms at all. And we want to know if there's something in my genetics that's different from Ryan's genetics. So I'll sequence my genome and I'll sequence Ryan's genome, and we've got it here on the screen, or at least a part of each of our genomes on the screen. And if I wanted to, as an individual person, figure out if there's something different in my genome compared to Ryan's genome that would answer this question or speak to this hypothesis, I would have to look at every single one of our A's, T's, C's, and G's in our genome. So that would take an awfully long time for one person to do, especially because there's 3.2 billion bits of these pieces of information in my genome compared to Ryan's genome. So to go through 3.2 billion bits of my information and compare it to 3.2 billion bits of Ryan's information, that would be an astronomical feat. It would take a really, really long time for any one human being to do. There's also another crux here, is that Ryan and I, despite our differences in appearance, we share a lot of our genome. So those A's, T's, C's, and G's, the 3.2 billion of them, are similar between people. I'm going to put this question out there, and feel free to answer in the chat. What percentage of the 3.2 billion base pairs in the human genome do you think you share with another human? So from 0% to 100%, what percentage of your genome, your A's, T's, C's, and G's, do you share with another random human? Nobody's answering in the chat. I'll give it another. Someone says 50% or greater than 50%. Any other guesses? 97, 98, getting closer. 99, Miles says. It's 99.9%. So Ryan and I share 99.9% of our A's, T's, C's, and G's. So it would be kind of redundant if we were to look at the amount of DNA that we actually share. So most scientists, genome scientists, are actually going to focus in on the 0.1% of information that is different. That still works out to be a lot. That's about 3.2 million bits of information. And again, if I'm just a scientist and I'm looking at these A's, T's, C's, and G's, examining my 3.2 million pieces of information with Ryan's 3.2 million pieces of information is still an astronomical feat. So we use computers to do this instead. We focus in on that 0.1% difference. And we look at that 0.1% difference as single nucleotide polymorphisms. So they're just variations in our genome. They don't necessarily mean anything. They're just variations. So I'm giving you an example here. In this symptomatic person, that's me, and the asymptomatic person, that's Ryan, we have these random variations between me and Ryan. They don't necessarily mean anything at this stage, but those random variations is where we're going to focus our attention. We're going to look at if those random variations are connected with a change in expression of symptoms. So that's where the genome-wide association study comes into play. Ryan, can you switch the slide, please? So the GWAS, or Genome-wide Association Study, will take those variable single nucleotide polymorphisms between a group of people that are symptomatic people and a group of people that are considered to be asymptomatic people, and we'll make those comparisons. So that here we've got our symptomatic group and we've collected their genome and their DNA in terms of those single nucleotide polymorphisms, those very specific 3.2 million pieces of information where we have variability. That's showing us, in this example here, we're seeing what those differences are compared to an asymptomatic person. And once we have that information, then we can start making connections. We can look at, all right, Ryan's DNA over here is different from Jessica's DNA. Is that difference located in a gene or a set of genes that may have an implication in terms of whether somebody has a symptom to COVID-19 infection? So what bioinformaticians have done is they've taken this astronomical feat of having to sequence 3.2 billion bits of information and made it 3.2 million bits of information, and given us places where we can look in the genome for an association to a gene that may explain the manifestation or the phenotype of expressing a symptom to this particular disease. Do you want to go to the next slide there, Ryan? Now, in order to do something so grand, because it still takes a lot of work and it still takes a lot of effort, and we're not comparing just one person's genome to another person's genome, we want to compare hundreds of symptomatic people to hundreds of asymptomatic people, and no one particular scientist or one particular lab can actually do that alone. Bioinformatics really is a collaborative effort. So the COVID-19 Host Genetics Initiative, which was identified in the article we're talking about, is doing exactly this type of research, collecting data from around the world to make it a collaborative effort of research scientists that are located in China, in France, in Canada, in the United States, collecting symptomatic patients and asymptomatic patients' DNA genome sequences so that bioinformaticians can start to look at those sequences and identify differences that could answer that question, why are some people symptomatic and other people asymptomatic? All right, that's all I've got for this topic. Let's open the floor to some questions. We've got one already. Are there differences between the two genomes of two people that are meaningless, i.e., are there meaningless bits within that 0.1% that do not matter? That's a really good question. And I'm not sure if you're familiar with the term junk DNA. People used to say a lot about junk DNA. So at one point, it was thought that the vast majority of our DNA actually doesn't encode for anything from a gene perspective. And instead, it's just sort of random bits of information that intersperse within genes. And the genes are the important ones. The genes give us information about how to make proteins, and proteins go on to give us function. So everything in between was called junk DNA. And in that junk DNA is where we find that variability, right? There are single nucleotide polymorphisms, there are telomeres, like you said, and there are repeated sequences and all kinds of really interesting stuff. So yeah, that's where the variability is. But that variability doesn't necessarily mean it has no meaning, because within that junk DNA, that supposed junk DNA, there's also sequences that are responsible for regulating how genes are expressed and regulating RNA, and RNA can have lots of different functions in our body as well. So to say that it's meaningless, it could not interfere. Sorry, let me say that again. It may not interfere with day-to-day functioning, but it could give rise to some of the information that is variable. So for instance, if that change is in an area of DNA that is a regulating element, then it could turn genes on and turn genes off. Or say, for instance, it has to do with melanin production in your hair. And if it's turning the melanin production genes on, then you get lots and lots of deposition of melanin in your hair, in your eyes, and your skin. Versus if it's turning it off, as is in the case in some folks, then you get less melanin production. I hope that somewhat answers the question. Ryan, do you have anything to add?
No, that was pretty much what I was going to go with, but way less biology sounding.
We've got a question from Brian. What is the meaning to the height of the bars on the graph for the Host Genetics Initiative website? Good question. If you want to go back to it. So this is a Manhattan plot, if I'm not mistaken, and the dots represent the expression of a particular gene on a particular chromosome. The higher the dots, the more expression level there is.
Yes. So if you would look at, let's just say, that TLR7, you would see a big jump in the X chromosome and the less jump in like your Y chromosome if you were looking at it for like a simpler one. Did I get that right?
Yes, yeah.
Perfect. Nailed it.
There's a lot of different ways to express this graph, but it is a really common type of plot to use when you're looking at sequences. So along the bottom, you might see ATCG, 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 and then each dot represents like a concentration of people that would have an A at that site, or that sort of thing. So there's a lot of ways to use a graph like this, which can be really helpful in identifying differences between people or places where to look.
Any other questions? Going once. Going twice. Okay. Sold. Okay. So if you do have a question and you think of it when we're doing our other talk, feel free to put in the chat or bring it up again during the second question period if you want. Okay, but we will continue on, and I believe it is Jessica again. Do you want control over the screen, or do you just want me to say when you want me to turn this?
Just give you a go ahead and click kind of thing.
Got it.
So most of you that are here probably are aware that we're offering a brand new program starting fall of 2020. It's a Baccalaureate of Science in Bioinformatics, and we're super excited to offer this for a number of reasons. It'll be one of the first baccalaureate degrees at Langara in the sciences. It is the first, actually. And this four-year program is in a very incredible and innovative new field in bioinformatics. So we're super excited to take on this new adventure with our students in mind. Next. So we've somewhat got the gist of bioinformatics, I think, from our earlier talk, and a lot of you are here because you're interested in bioinformatics already, but I just wanted to give a quick definition of what bioinformatics means. I like to think of it as the marriage of disciplines, particularly biology with computer science and math and statistics. So the idea here is that somewhere along the line, probably around 2000 or 2002, biology became a data science. And what we mean by that is that through human genome sequencing or genome sequencing in general, we realized that every cell in an organism has a lot of information associated with it in the form of DNA. And those are the A's, T's, C's, and G's we showed you in a previous slide. And that in order to make sense of that information, humans themselves cannot analyze that amount of information alone and make any kind of sense of it. So we had to co-opt computer science and math and statistics to be able to manage and analyze and make sense of that data, to do things like new discoveries or problem solve or answer really complex biological questions, such as why are some people symptomatic and other people asymptomatic when it comes to COVID-19 infections? Or through the use of personalized medicine, how can we think about somebody's background genetics to be able to develop brand new diagnostic or treatment strategies for an individual person using that information? So bioinformatics has given biologists a whole new tool in their toolkit in order to be able to do a better complicated research and manage that big data. Next. So the question becomes, why would we want to study bioinformatics? Why would you want to do a 4-year degree in bioinformatics? And the quick answer is that the market is growing substantially year over year. We're seeing a lot more money being put into this discipline and into this industry. So for instance, the bioinformatics market was expected to grow at about $13 billion by 2020, and in fact it did. We also saw that more money is being put into this area, including to pay for labor and jobs. So when we look at starting salaries for a bioinformatician that's coming out with a baccalaureate degree, the starting salaries are quite high, higher than other sort of life science, chemistry, physics types of entry-level positions at about $47,000 to $53,000 per year. And what we've also seen over the last 10 years or so is the number of jobs that are created every single year are increasing exponentially. So in the United States alone, the Department of Labor projected that 3,600 new jobs would be created from 2016 to 2020. That's within a 4-year period that had the label of bioinformatics specifically. And that the total employment in 2016 was about 39,000, and it has also grown exponentially if we look at it today. So there's lots of jobs in this area, and there's certainly a very high market value for somebody that is just starting out in a career compared to somebody that has a baccalaureate degree in biology or a baccalaureate degree in chemistry. Next slide. So the last question that I'll deal with, and then I'll pass it along to Ryan here, is why would you want to take a baccalaureate degree at Langara, particularly in bioinformatics? And I think there's a lot of reasons to do this. Langara is a great school, and we've put a lot of effort and emphasis in putting together a program that truly is innovative and collaborative and applied. So when I say that this is a collaborative degree, I mean that the Department of Computing Science and Biology and Chemistry and Math have worked together to create this curriculum because it is a multidisciplinary, interdisciplinary field. So we've worked together internally to create the curriculum for this program. But we also worked very much part and parcel with our industry partners around the Lower Mainland. So we worked with UBC, we worked with SFU, and we worked with all kinds of different companies, including Molecular U, Genexus, to create a program that would be valuable for our students in getting a job at the end of the day. So it's been a truly collaborative effort, and we've had the support of the big universities in town, as well as our industry partners to put this program together. It is hands-on and applied right from the beginning. So when we do courses at Langara, particularly in the sciences, we're looking at having each of our courses has a lab component to it. So you're going to be either in a wet lab or in an in silico dry lab, a computer lab, doing work that's real world based problems or hands-on and authentic type of learning. We also have incorporated in the curriculum, which Ryan will talk about in a few minutes, capstone projects where students can work on their own projects in their later years. And then finally, there is an 8-month co-op piece in which you will go out there and you'll get industry-related experience, again, an applied and hands-on approach to learning. So this truly is an innovative, applied, and collaborative type of program. You won't find it at any other university. There's no baccalaureate degree in bioinformatics at UBC or SFU. This is truly a unique experience that Langara has created as a collaborative effort across disciplines and across institutions in Vancouver. Right? And back to you.
Yeah, just have to unmute. Okay, so let's talk about the program as a whole and kind of the emissions. So right now we have 2 emissions kind of being set up. We have students who are entering first year and students who are entering third year. So that being said, so people are entering here and people are entering around here. I say around here because if you're missing everything, but you have most of this stuff completed, still apply. We can probably still get you into a third year type of start, even if you don't match everything perfectly. But so this program's designed for bioinformatics. So this, you're going to come through knowing a lot about biology and a lot about computer science. Because if you look, all the green courses you see, those are biology courses. All the purple courses, those are computer science courses. And then the blue is going to be your chemistry, and the orange is going to be your math and statistics. So you're leaving here with a lot of experience in all of these fields that you need to be effective in the industry. Now, coming from someone like me, I have a degree in computer science, but I am super interested in biology. And so anytime I tackle a new problem, I dive in, I learn all the knowledge I need to know, and then I go and I try to apply that. Someone coming out of this program wouldn't have to do that because a lot of that background knowledge I always have to bring myself up to anytime I'm solving a problem, you know, because you've learned it somewhere along the line. That actually came up with the presentation we did. I learned a lot about these receptors and how they work, and it was super cool. Yeah. So this was also designed to be programmed to be a very practical program, because if you look, all of these courses will involve a lab component as well. So not only are we going to be teaching you a bunch, you'll also be getting a whole bunch of hands-on experience, really getting familiar with how all this stuff works. Okay. And a lot of these courses, so everything you see kind of with the border, these are going to be new courses for the program. So these are designed with mind. We talk to the industry. These are the courses they think are important, and we develop them that way. Even some of the courses early on are getting reworked. So for example, if you're entering as first year and you take 1150, that course is traditionally taught in Java, but we're going to play it up and we're going to teach you Python because that's what the industry wants. So a lot of good stuff. You leave with understanding of both comp sci and biology and enough math in there to make you very effective in this field. Okay, so one thing about employment, so we have one of those jobs. So type of jobs you'll look at getting would be research technician, developer, system analyst. All of these jobs are what you're going to be seeing when you walk away with this, because sure, you're going to get a degree in bioinformatics, but you're also going to have the skills to understand how to interpret data from a natural science, which is a very useful skill to get. So could this be applied to other sciences as well? Yeah, you'll have that, but you won't have as much background in the science you're applying to, but you'll have that knowledge with you. So a lot of these the analyst jobs, you'll see a lot of that come up. Okay, so here is for the part of the soft sell where, so you might commit, and that is great. You come through this program, you graduate, you now have a bachelor's of science in bioinformatics. You can take that to the industry, get a job, and live your life perfectly happy. You can also take it, because it's a bachelor's degree, you can also take this into graduate studies. So if you're like, this is really neat, I'm passionate about, I would love to do this more, nothing's stopping you from doing this and going into a master's degree and a PhD from there. So this isn't the stopping point to graduate. It could be, but you can also go more. Say you take the courses and you end up completing the first 2 years, but it ends up not really being for you. Well, we also have the associate and the diploma degree. And by completing this program, you have most of those courses already done. So in 2 years, this isn't for you. Finish up whatever you're missing and then graduate with the associate or diploma. That's perfectly fine. And say you enter first year, you get into the program, you're like, I love this, let's do it. You take biology or you take computer science, not for you in any way. Don't worry, all the courses you took can still be transferred to another university. You can take that, enter another actual program at Langara. It's all transferable from that point. So there's lots of ways you can always leave, continue on, or there's other parts along the way. So it's not like you're buckled in. This is a 4-year program. If you don't complete it, you get nothing. There's lots of ways to branch off. I forgot the most important thing, co-op. So with this program, sorry, I was leaving. With this program, you also will leave with 8 months of working experience. So we'll help you get a co-op job, and you'll leave that, and you'll get actually working experience, which is kind of really valuable because most jobs you look for always says they're looking for someone with experience. You'll have experience by the time you leave this program. Okay, and that is all I have to say. So we're going to open up to questions again.
Well, Phoebe had a really good question already. How would AI influence bioinformatics? And she's saying, oh, I think influence goes both ways. And in fact, it does, because AI can help with pattern recognition in a way that humans are not capable of. We use a lot of AI when we're doing, or machine learning, I should say.
Thank you. I was going to correct you on that.
Yeah, we use machine learning when we're doing when we're looking at tumor slides in our lab. We've designed it so that machine learning can identify different markers within lots and lots of different cells within a tumor, where a human wouldn't be able to see that kind of thing. So certainly there is a huge role for machine learning in bioinformatics in the future, and currently and in the future. And I'm not sure if you noticed, but in the curriculum, there is a machine learning course in 4th year.
That's actually what got me into bioinformatics in the first place, was machine learning algorithms.
Mm-hmm.
That's what got me interested. It's a hugely important thing. And it
Mm-hmm.
frustrates biologists beyond belief sometimes, because they were like, hey, I would like you to find out what this data means. And you're like, hey, it means this. They're cool. What's happening to make that decision? You're like, that's inside the box. We don't know what's happening. Because neural networks they make all these connections that are completely, you really can't understand them looking
Mm-hmm.
at it, but the machine picked up all these decisions. So
Yeah.
there's lots of machine learning that goes on in bioinformatics. Yeah.
We have a question. If I am already in the associate degree, is there an entrance GPA requirement for 3rd year? For the program, you need to have a 2.6 GPA in order to get into co-op. So the entry requirement for 3rd year would be a minimum of 2.6, but it's not an official entry requirement. Are there any other questions or comments? Is it not sufficient for students who graduated with a diploma or associate's degree to go into the job market? It would be much more difficult. So this program we put together with the employers. So we had a lot of bioinformatics employers that were involved with the curriculum design. And even they suggested that a 2-year program is not sufficient and that a 4-year program would be much better. So while there's a pause, I wanted to introduce that Reese is here as well. You can see her on your screen. She's a recruiter working in the registrar's office. So if you do have any technical questions about admissions or applications, she can answer those for you as well.
Thanks, Jessica. Hi, everyone. Nice to see everyone here. So I do have one update to share with you guys. So now we are still accepting the application for the full semester, and the application deadline will be until June 21st. So if you're interested in this program, please go to our website to double check the prerequisite requirement and academic requirement. If you have any questions, you can feel free to contact us directly.
Mm-hmm. Reese, do you want to put the website in the chat? Thanks.
Yeah, sure.
What is the bioinformatic research focus at Langara? That's a great question. We've got a number of different projects on the go at the moment that use bioinformatics as a tool, and it's growing. So Langara's applied research program is still fledgling, but so right now there's a few research scientists. One group is working on DNA sequencing of ants, if you're interested in that. There's another group that's working on sequencing hops that are used to make beer. So there's some Vancouver varieties of hops that are used in the microbreweries very commonly around the Lower Mainland. And we're interested to know if the DNA and the chemical constituents of the hops are influencing the flavors of beer. So we've been working with 49 Parallel. I think that's the name of the brewery. Parallel 49. Yeah, I couldn't figure out. Yeah, there's a coffee shop that's the other way around. But Parallel 49 is the brewery. And at one point, they were actually selling a Langara brand beer, which was really fun for us. And then the last project that's just starting to get off the ground, we're looking for funding for it, is a cannabis project. Similar to the beer project, we're looking at whether the sequences of cannabis, cannabinoids in cannabis, as well as the chemical constituents, play a role in the function of cannabis as a possible treatment to different diseases. So you may have heard that people use cannabis for pain management, for sleep, for inflammation, for all kinds of other things. And what Langara is interested to find out is whether the DNA sequence of the plant plays a role, and the chemical nature of the plant plays a role in its efficacy as a treatment for these types of indications, including pain and for sleep and for inflammation. So that's sort of the gist of the projects that are currently ongoing at Langara. Any other questions? Are there any interests in comp sci? I know that Ali Reza and others are thinking about doing some bioinformatics work.
I don't think we currently have anything that's going on. Is that correct, Brian? Not that I know of. You got a nod. Any other questions, comments, concerns out there? If not, we can call this a session. And yeah,
What will most of the projects look like? As in, is it statistic and data processing?
it could be. That's the whole thing. We don't, like the capstone projects, we don't know what those could look like. A lot of that could be pulled from whatever co-op you have. You could be working on a project and you might be finishing up there. And that could be anything from writing a new machine learning model to using previous existing one to do some sort of data analysis of it, or even wet lab stuff, collecting samples, running it through. So the projects can really run the gambit of this discipline. Because there's kind of three parts to bioinformatics. You have wet lab stuff, you have the computer science, so writing these new algorithms, figure out that. And then you have this pipeline in the middle where, okay, getting from point A to point B, putting all the right tools in the middle. And that's kind of the three parts of bioinformatics. So I like to think about it. And you could be involved with any one of those. Any of those could lead to a project.
Mm-hmm. There's a
Does
question
that answer the
about
question? So the answer is we don't know what they're going to be.
there will be some variability. And some
So
of those projects will
froze.
come
It's
from the
froze, Abby.
instructors that are currently doing research. So there's quite a few of us in the biology department that are doing wet lab exclusive research, and then some that are doing more of the bioinformatics side of things. So it really depends on the student and what the student is interested in, and it can go in a lot of different directions. We have a question from Sean. Are the admission requirements lower for the associate's degree and diploma than the degree? Yes.
Yeah.
Go ahead.
Oh, sorry.
Ahead,
Yeah.
Brittany.
So associate degree and diploma are open enrollment programs. So the student will be required to graduate from high school, either BCE grade 12 or the equivalent, or be 19 years old or older, they will be admitted. But for the bachelor's degree, there are a few different prerequisite requirements the student needs to meet. So I would recommend you to visit our website to double check the academic requirement for the bachelor's degree. There are a few different prerequisites.
Brahim, not that I know of at the moment, but we could certainly create a connection with people. Sorry, the question I should reiterate is somebody has a very specific interest in epigenetics. And at the moment, there's no epigenetics work that's being done at Langara. But the nice thing is, because we have these connections with industry partners and academia, is that we could build a type of research project that you could do, and we would pull on these other folks that were interested in the field. Thanks. Can students transfer to year 3 of the degree after completing the associate's degree or the diploma? So that's a bit of a complicated question. The first time we offer this in fall of 2020, you can enter in as a first-year student or you can enter in as a third-year student, but it's the only time we're going to officially offer a third-year entry. Going forward, however, if there's space in the program and it happens to line up that you finished your first 2 years and there is a third year that's happening, then you may be able to backfill the program, but there's no promises there. There would have to be space, and it would have to align with your graduation from the 2 or 2-year degree or diploma programs. Does anyone want to add to that? Maybe.
That was what I assumed, but I think Brian might have.
Yeah, Brian.
I was also going to add in that, many of the third-year courses only have prerequisites based on the specific courses that you have taken in your second and first year. So if a third-year course is available and you meet the prerequisites, then by all means, go ahead and register.
Mm-hmm. And then there's a question, after this intake, when is the next intake opportunity? We hope to be offering first year every fall from here on in. So the next intake would be fall of 2021. Good questions, everyone. I'm going to plug in the email address for the program. If you think of a question that you didn't have now, please feel free to send it to bioinformatics@langara.ca, and we'll be sure to answer it. Or send it to the right person to answer.
Okay, I think that's everything. So if there, oh, wait.
So Phoebe, if you do want to see examples of projects, I think the best thing to do would be to talk to the instructors for the courses in which the projects you're like if you're thinking of labs, for instance, then it would be to talk to the instructor for the course. If you're interested in research projects, then I would put you in touch with the person who's doing the research, because there is a wide variety of projects that are available. And it might also depend on where you do your co-op opportunity. So if you're working in the BC Cancer Center, then the projects will be very specific to cancer. If you're working instead in molecular U, then the project will be more oriented towards in silico stuff. So it's really hard to say exactly what the project would look like. But you will be well supported. When will applicants find out if they've been accepted to the program? So we're doing rolling applications, which means that as long as you have all of your materials in and we can assess your materials appropriately, then you'll get a conditional acceptance. Sorry, conditional acceptance if we don't have everything, but an acceptance to the program as soon as we can assess your application. So if you submitted an application yesterday, give it about a week or two for our registrar and enrollment services to get the information to us. We'll look at it and then we'll send you an acceptance if it sees, or if it fits the bill. So it can happen really quickly. Is there a list that I can look into these co-op overseers? Not quite yet. Co-op is still a distance away. So if you are entering into third year in fall of 2020, the co-op term won't be until summer of 2021. So that's way too far in advance for co-op jobs to be available at this time. But I can give you a list of companies. In fact, I think I had a really good slide, not on here, but there's a lot of different companies that have suggested that they would take our co-op students. So if you send an email to bioinformatics.ca, just with that, or at langara.ca, send that question and we can maybe share with you some of the companies that you may be working with. You know what I'll do? I'll put that online somehow on our website. I know Ryan's hesitating to say, if there's nothing else.
If there's nothing else, well, every time I say it, like 3 more questions pop up. So if there's nothing else, if there's no other further question, do you have a question, feel free to ask. We are more than happy to answer anything you might have. But if you don't want to do it here, do send it to the email. That would be a good way to get a hold of us if you wanted to. Yeah. So if you don't have any questions, ask it now. If you want to ask it later, feel free to send it to that bioinformatics@langara.ca email. That's somewhere in the chat above.
Mm-hmm.
And we'll either answer it or direct you to somebody who can answer it.
I think that's enough of a pause now. We can say thank you, everybody, for spending your time with us today,
Yes.
this evening. And we hope to see you in the program.
Yeah.
You're welcome, Miles. You're welcome, Kai. Thanks for your good questions, everyone. Have a wonderful evening.
Enjoy Tuesday? Is it Tuesday?
I'm going to stop recording too.